Hello students, welcome to today's lesson on antibiotics production. In this lesson, we are going to study antibiotics and how antibiotics can be produced. Antibiotics are chemical substances or pharmaceutical drugs that can inhibit the growth of germs and even it capable to damage or kill germs or bacteria. The first antibiotics were synthesized from fungi. The great scientist Alexander Fleming discovered the first antibiotics, penicillin, in 1928 from a bread mold type, Penicillium notatum. There might be a certain bacterial disease that we might experience in our community. Here are some examples of bacterial disease that we might experience. The first one, cholera. This cholera can be caused by Vibrio cholera and it can be treated through antibiotics like doxycycline and ciprofloxacin. The second types of bacterial disease that we might experience in our community is typhoid fever, which is caused by Salmonella typhi and can be treated by chloramphenicol, ampicillin, and amoxicillin. The third types of bacterial disease that we might experience in our community is tuberculosis or TB. This disease is caused by Mycobacterium tuberculosis and it can be treated by using antibiotics like isoniazid, rifampicin, and pyrazinamide. The fourth types that we might experience, typhus, which is caused by Rickettsia prowazekii, and this can be treated by using tetracycline, doxycycline, azithromycin, and rifampicin. The fifth type that we might experience in our community is trachoma, that is caused by Chlamydia trachomatis, and it can get treated by azithromycin and tetracycline. These highly valuable antibiotics are increasingly being made by using genetically modified bacteria in huge fermenters. Antibiotics in special fermenters can be produced through fermentation processes. These fermentation processes to produce antibiotics involves the following valuable steps. The first one, we need to isolate the microorganisms or bacteria that can produce antibiotic that we exactly require. After we get the bacteria that can give us the antibiotic that we demand, we need to culture them in laboratory on nutrient medium. In laboratory on nutrient medium, they can reproduce and grow for a certain days. After then, we need to transfer or shift these cultured microorganisms or bacteria into seed tank. Why we need to transfer or shift them to seed tank? What is seed tank? Seed tank is a small tank or fermenter that is designed to keep well-defined strains of cells or bacteria which is viable at high density for further upstream processing or fermentation in bioreactor or huge fermenters. So since seed tank had the same condition as huge fermenters, it helps microbes to adapt the condition that they will face in huge fermenters. So this seed tank is highly valuable to train these cultured microorganisms before they added into special huge fermenters. Then we need to transfer or shift them into the fermentation tank to yield antibiotics. In these special fermenters, they get a constant suitable environmental condition. Since they acquire all the required things in this special fermenter, they grow, reproduce, and produce antibiotics. These antibiotics are bacterial or microorganism byproducts or metabolites. But these antibiotics or these metabolites are highly valuable to treat germs that can cause disease. So to have these antibiotics, we have to isolate, purify, or refine these antibiotics from other non-valuable metabolites or impurities. Here is the structures of special huge fermenters. Structures of fermenters for antibiotics production. These fermenters are mostly made from materials which cannot be corroded, such as stainless steel or special alloys that can resist acidic wastes of most microorganisms. Since microbes produce acidic wastes that can corrode ionic materials. So this special fermenter has inlet pipe. Here it is. This inlet pipe is the passage where all the nutrients and cultured microbes passes into the fermentation tank. Another is a steam inlet. This steam inlet is the passage in which steam water gets added into the fermentation tank, since microbes need the temperature at optimum level. Another part of this special fermenter is temperature and pH probe. This fermenter has temperature and pH probes. These temperature and pH probes are indicators that inform the levels of temperature and pH. Then, by having this information, we can adjust temperature by adding cold water into the water jacket, and we also adjust pH by adding alkali or acid into the fermentation tank. Another part is cold water inlet. This is the pipe that lets in cold water into water jacket to maintain constant temperature of the fermenter. Another is water jacket. Here it is. This is the cavity outside the fermenter in which cold water gets stored for the purpose of maintaining temperature constant in the fermenter. Since fermentation produces heat, so to prevent the contents from overheating, the fermenter is surrounded by a water jacket through which cold water circulates and keeping the temperature at the best level for growth. Another part is, here it is, air inlet. Air inlet is the passage for filtered air. If the microbes in the fermenter respire aerobically, this air inlet mostly has filters. This filter is responsible to filter out the air to prevent contamination. Another part here is steam outlet. This steam outlet is the pipe in which steams leave out. Another part is, here it is, exhaust outlet. This exhaust outlet where exhausted gaseous wastes of the fermentation escape or move out. Another part here is cold water outlet. This cold water outlet is the passage where cold water moves out from the jacket when temperature of the fermenter gets low to the temperature constant. Here is a motor. This motor is a device that spins the steering paddles to mix the contents of the fermenter. As you can see here, there is also this steering paddle. This steering paddle, when gets spun by motor, it mixes up the contents, so simply keeps the microorganisms in suspension in the liquid. So these microbes or bacteria get more exposure to the nutrients and helps to keep the temperature or constant throughout the vessel. Some fermenters use jets of air to mix the contents instead of paddles. So, genetically modified bacteria are highly valuable or important to produce insulin hormone, human growth hormone, antibiotics, enzymes for washing powder, human vaccines such as vaccine against hepatitis B, and others in special fermenters at industrial levels.